Okay, so digestion. The alimentary canal of a mammal is a long tube running from one end of its body to the other, that is from the mouth to the anus. You can see diagrams. Okay, you can see diagrams from the internet, or I'll upload for you. Before food can be of any use to animal, it has to get out of the alimentary canal and into the bloodstream. This is called absorption. Why we eat food? So that it can be respired, and respiration is done by the cells. And in the alimentary canal, the cells are all over the body. So from the alimentary canal, they need to be transported to the cells. How are they going to be transported to the cells? They will be transported via the bloodstream, the blood vessels. And so from the alimentary canal, the digested foods needs to be transported to the to the bloodstream. The process of moving the digested food from the alimentary canal to the bloodstream is called absorption. And by the bloodstream, I meant the blood vessels. Okay. To be absorbed, molecules of food have to get through the wall of the alimentary canal. They need to be quite small to be able to do this. The food that is eaten by animals usually contains some large molecules of protein, carbohydrate, and fats. Before these molecules can be absorbed, they must be broken down into small ones. This is called digestion. So before absorption, digestion needs to take place, where the large macromolecules are going to be converted into small molecules, so that they are enough small to be to pass through the wall of the alimentary canal. As you know, the walls do not allow all molecules to pass through. You will learn how the selective transportation of molecules take place from your chapter in your A levels, where you will learn about in detail about the structure of the cell membranes. Figure 7.10 shows what happens to the three kinds of nutrients that need to be digested: fats, proteins, and carbohydrates. Look at one of a time and work down it to follow what happens to the type of food as it passes through the alimentary canal. Large carbohydrate molecules such as polysaccharides have to be broken down into simple sugars. So simple sugars can be passed through the alimentary canal, and simple sugars are monosaccharides. We have known that monosaccharides are the kind of carbohydrate that contains only like 1 unit of the carbohydrate, like glucose. Okay. Proteins are broken down to amino acids. Fats are broken down to fatty acids and glycerol. Simple sugars, water, vitamins, and minerals are already small molecules, and they can be absorbed just as they are. They do not need to be digested. Look at this table here. Mechanical and chemical digestion. Now there are two types of digestion. Often the food an animal eats is in quite large pieces. These pieces of food need to be broken up by teeth and by churning movements of the alimentary canal. This is called mechanical digestion. So first of all, mechanical digestion, no chemical reaction occurs. You are mechanically digesting it with your teeth. You are breaking it down, and then you are making it into a bolus. Okay, a bolus is like this. You make the food into a ball shaped like this when you eat, and then you swallow it. There is a rhythmic movement called peristalsis, which cause the food particles to become shaped like this, so that they are easily transported through the esophagus. This part is called the esophagus, the tube which connects the mouth to the stomach. Now, for the peristaltic movement, I want you to watch a YouTube video. I want you to go to YouTube and search peristalsis, and watch the animation. It is basically the rhythmic contraction and relaxation of the alimentary canal so that the food particles can pass through its way. Now, one piece of food must have been ground up, and once piece of food have been ground up, the large molecules present are then broken down into smaller ones. This is called chemical digestion. So in mechanical digestion, none of the macromolecules is changed, but after they have been broken into smaller pieces so that the surface area can increase for digestion, chemical digestion, the macromolecules are changed to smaller ones so that they are monomers. So this type of digestion requires chemical reaction, and thus it is called chemical digestion. So why mechanical digestion is needed? So if a food particle was this big, it would have a large surface area, and so the rate of chemical reaction would be slower. But if the particles were smaller, it would have a small surface area. But if the particles were smaller, the surface area would increase, and thus the rate of chemical reaction will increase. You will learn about how the surface area affects the rate of chemical reaction in your chemistry of your O levels. So larger surface area equals to more rate of chemical reaction, and if for big pieces, smaller surface area, and it causes lower rate of reaction. Okay. It involves a chemical change from one sort of molecule to another. Enzymes are involved in this process. Okay. Now, mechanical digestion, key definition: the breakdown of food into smaller pieces without chemical change to the food molecules, and chemical digestion, the breakdown of large insoluble molecules into small soluble molecules. And you will also add like enzymes are involved. Okay. Now let us see the diagram. Okay, fats, proteins, and carbohydrates. For fats, teeth break down pieces of food into smaller ones. Yes. Bile cells break down large droplets of fat into smaller ones. Now we came across a new term, bile salts. What are bile salts? Bile salts are produced from liver, but they are stored in the gallbladder, and then they are excreted in the they are not excreted, they are they are introduced to the alimentary canal through the duct, the gallbladder. Okay, the gallbladder is not the duct, but the gallbladder stores the bile salts, and there is a duct through which the bile salts, that is the bile which contains the bile salts, are transported to the alimentary canal. So bile salts breaks down large droplets of fat into smaller ones, thus increasing the surface area. Now if I ask what is the function of bile or bile salts, you have to answer that they break down large droplets of fats into smaller ones. Fat droplets. So we have fat molecules. Now lipase, the enzyme that breaks down fat, lipase breaks down fat molecules to fatty acids and glycerol molecules. Now for protein, teeth break down large pieces of food into smaller ones. Water in digestive juices dissolves some food. Okay, so water dissolves some food in digestive juices. We all know what are digestive juices. Digestive juices contain the enzymes. Okay. Now, small pieces of food and some food in solution. We have protein molecules. Protease breaks down protein to peptide molecules. Well, protease is an enzyme that breaks down protein into polypeptide molecules, and peptidases breaks down polypeptides to amino acid molecules. So here we have come across few terms: polypeptides and protein polypeptides and amino acids. Okay, let me explain. You will learn this in your macromolecule chapter, the biological molecules chapter of your O levels. Say for example, if this is an amino acid, okay, containing an NH2 group and a carboxylic group. This structure in detail you learn in your chemistry and in your A levels. So this is an amino acid. If we have, say, two amino acids joined together, they form a peptide. If we have maybe 3 or 4 amino acids, they are called peptides. So few amino acids joined together is called a peptide, and this is called a peptide linkage. But if few of these peptides join together to form a large chain of amino acids, so we have two peptides. It is called a protein molecules. So amino acids, peptides, and this is many peptides joined together to form a protein molecules. So a protein molecule is also a polypeptide. Okay, fine. Now let's go to carbohydrate. Teeth break down large pieces of food into smaller ones. Water and digestive juices dissolve some food. Okay, fine. Small piece of food and some protein in solution. Starch molecules. Amylase breaks down molecules down to maltose. Maltase breaks down maltose down to glucose. Okay, so for starch, amylase breaks down starch to maltose molecules. Maltose is a disaccharide and amylase is a polysaccharide. But our main intention is not to produce a disaccharide but to produce a monosaccharide so that they can pass through the walls of the alimentary canal. So then maltase breaks down maltose to glucose molecules. Okay, very easy. Now if I tell you, write down the conversion of carbohydrate to glucose with all the enzymes involved and how this or that happen, you'll simply say that teeth break down large pieces of food into smaller ones. It is common for everyone. If you wish, you can give water and digestive juices, dissolve some food, but it is not so much necessary. But what's necessary is you give amylase breaks down starch molecules to maltose, and then maltase breaks down maltose to glucose. Like this for fats, proteins and carbohydrates. You need to learn by heart.